in the morning, we continue with problems which are related to active matter. And in that case, we'd like to just to start a little bit with some introduction. And I will start just by showing, we'll do it with my hand. Wait. Okay, so what I would like to start with is to talk a little bit about places where you see in nature buckling effects. So here are about two examples. So the first one deals with epithelia buckling during processes like gastrulation. So the gastrulation is a process when you have cells or tissues that form into embryo. And the process of this formation of this tissue into an embryo is related or induce some buckling of the tissue itself without anything that is related to cell division. So this is an example. And in that case, what we have usually is cells which are connected one to the other via cell adhesion. And this is one thing. And below it, usually they are attached to a substrate which can be elastic. And it was proposed that these buckling effects are related to the fact that you have contractile forces which are generated inside the cells, cell-cell contact, plus the elastic interaction with the surface. And one of the things that we would like to ask in this talk is whether contractile forces are really sufficient to generate buckling, and without the need to any interaction with any substrate or any interaction between cells. So this is the thing that we are going to talk today. Another example where you see buckling in nature is related to flowers and leaves, which is more related to what Eran did. And in that case, it was shown that in the case of differential growth process, which means that the boundary can grow at a different rate than the center, in that case it is growing faster, you can generate instabilities at the boundary of the tissue. And these instabilities, these buckling instabilities, can be regulated genetically, okay? And the question is, of course, whether these growth processes, these buckling instability, are related to any kind of non-uniformity in the material, at least in the lateral plane, and how they are genetically controlled. So what really dominates this type of effect? I don't think that there is really a clear answer about it. So what we are going to talk about today is contractile sheets, and contractile sheets mainly because we are interested in what is happening in tissue, animal tissues. So no plant tissues, animal tissue. And when we are talking about contractility, we can find contractility at different levels. So we heard about muscles, okay, about muscles. So we are talking about, in this case, contractility is generated by what we call myosin motors, which are working on actin filaments. Cell, this is an individual cell, can apply contractile forces on a substrate. So these are pillars from PDMS. You can really generate the forces generated by the cell, which is applying on the surface. The elements that in individual cells that are applying these contractile forces are, I would say, stress fibers. These are elongated fibers of actin and myosin, and the cell cortex, which is a very thin sheet, okay, of network of actin and myosin, which is really lies below the plasma membrane and really can generate this, for example, this process of division. So these are the places where you can see these contractile systems, and we are interested mainly in those type of effects. So just a very, very small reminder, what is important to generate these contractile forces is the fact that we use chemical energy in the form of ATP, and we transform it into mechanical work by using what we call molecular motors. So this is an example of a molecular motor. These are actin filaments which are found in the cell cytoskeleton. These are polar structure. In fact, These molecular motors, this is only the head of the motor, this is the tail of the motor, is really utilizing every movement of this motor along this track. This is what we call utilize one ATP molecule, and in this way can move along this track. Now, how do you generate contractile forces is the fact that you can use, for example, in that case, we give an example where we have two opposing actin filaments. So I told you they were polar. So we have two opposing direction of polar filaments, in that case actin. And you use, instead of using one molecular motor, you can use aggregate of molecular motors. So you have multiple head structure, and each of these heads interact with another filament. And since these heads are moving in a given direction, in that case what we call the plus end, they can really generate relative motion between the two filaments and effectively generate contractile forces. Now, the point is that if you use big aggregates of motor and many, many filaments to generate a network, in that case, you generate really a contractile network. So what we do in the system, we use actin filaments. We use another molecules that stabilize this filament to generate what we call bundles. So this is the way the system is schematically looking like. So we have aggregate of motors that can interact with several filaments. You can imagine that you have many more filaments around. Okay, they generate this type of, you mix everything all together inside small chambers. The idea is that the chambers that we're using are very thin and relatively wide. And in this configuration, we will be able to generate thin elastic structures that can contract. So this is the idea, to generate contractile sheets. So when you mix everything together, and we don't mix filaments, we mix really the monomeric structures that generate these filaments, the system will polymerize and form a network and eventually start to contract. So this is really the onset of contraction. I don't show you yet the contraction process. Okay? And if you look at the structure of the network, so here what you have is this gel, this actin gel that is in green. And what you see here are these the molecular motors aggregates that we label in a different color. And they are here. They are situated in the network. If you, what happened is that in fact the motors are really localizing at these apex, really, and they are really positioned inside the network. And this is basically, if you do the correlation, density correlation function, you can really see that you have a liquid-like structure, but the motor really are embedded inside. If you prepare such a network, such a gel, and only afterwards add these contractile elements, which are the motors, nothing works. I mean, they can walk on top of this network. You can see movement, but no contraction. So everything should be prepared all together and at once. Okay, so we generate this type of network, and now what we do basically is, so we have these very, very thin chambers. We put a drop of this solution, we let the system polymerize, and after it finishes to polymerize, it starts to contract. So this is basically a drop of this solution. It polymerized, I've shown you just before, you get a very nice network. And now we zoom out, okay, and look from things from very far, in order to be able to see and to localize the whole system. And what you see is that the system contracts. Okay, this takes 2-3 minutes. I mean, it's an extremely efficient structure, and it contracts to something like 90% of its original size in 2-3 minutes. It's extremely efficient. Okay, we cannot stop it. So this is one of the problems that we have. We cannot slow everything down. If you try to slow it down, so everything changes. So this is too optimal, I would say, in many sense. So what? The fact that you have ATP in the system and the motors can interact with the filament, the minute they start to form, they start to move on top of each other. And I will have a network. The motors interact with the filament in an irreversible manner. They cannot apply any force. So it is, I would say, a frozen gel. So nothing will happen. They will polymerize, but nothing will happen. Okay, so you need really this ATP to generate. Une seconde. Wait. So So what happened basically that during this contraction process we can really measure, not here of course in the precedent image that you saw, you can really estimate the mesh size. So what we see, we look at the holes, we estimate some certain mesh size only to have an estimate of the system contractility. And then what we can do is really at different times to look at the distribution of these meshes, and you see that the system contracts. Okay. So it is important to say that in that case, contraction is induced by sliding of the motors along these bundles and not buckling. We don't have any buckling in the system. So if you use different type of I would say, cross-linking, passive cross-linking proteins, you can generate buckling. Here, it is really a sliding effect. So the bundles that are connected by motors at these different places are really only shrinking but not buckling. Okay. Okay, let's continue. So the question is, what drives this contraction process, and what is the mechanism? So the full mechanism we don't have yet, and it's something that we are trying to do with FE, but I will show you something that we can say about the system. So the first thing that we realize that contraction initiated the periphery. So we have two basic experimental evidence for that. The first one is that if you take the gel, so imagine this is the drop, and this is the gel from top, and you look how the density, okay, the active fluorescence, which mimics the density of the system, okay. If you look how the density distribute along this line, so over the whole gel structure, you see that initially, before everything starts, only in the beginning, after it polymerizes, basically the system is homogeneous. Okay, so these are the boundaries of the drop. Okay, this is nothing else. And now the system starts to contract, and you see very, very soon, just after the system starts to contract, that you have the densification, okay, of the gel at the boundary. So maybe we can go back. So it's not, let's see if it works like this. Like this. So it's not very clear to see it here. I mean, the resolution is really not optimal. But what we see that, although here it is less clear, if you take a little bit thinner systems, you really see that the density of the edges in the beginning is much higher than in the center. So this is what we see in here. Next, the second thing that we did is, in fact, to look at the distribution of the velocity distribution. So what you see here is an analysis of the velocity, the local velocity across the gel. So it is a little bit difficult to see, but what we see, and it is really from the beginning that it happens that we have the local velocity is maximal at the boundary and it vanishes at the center. Okay, so these two evidence give you some hint about the fact that everything starts from the boundary. Okay, we still need, of course, to, I mean, if we had a local or how the strain distribute inside the system, it would be better. But this is the first evidence, and this is clearly something that we can utilize. Now, what we did is, in fact, that what we can do is now to follow how the diameter of the system changes with time and extract from it the velocity of contraction of the gel edge. And what we see is a very characteristic behavior. So initially, okay, so this is the phase where I show you that the system polymerizes and generates the gel, etc., etc. And this is the point where everything starts to contract, okay? And what we see is initially a phase which is linear, which is go linearly with time. It reaches a maximal velocity, and then it decreases exponentially with a characteristic time tau. And this characteristic time is in fact related to the diffusion of the elastic stress of the gel during the contraction process. And we have two important, I would say, parameters that characterize this diffusion constant: the elasticity, okay, of the gel, of the elastic gel, and the friction. And friction is proportional to the viscosity of the medium, of the solvent, I would say, in which the gel is embedded, and the typical mesh size. What is important to note that the relevant length scale of the system, L, at this point is L max, okay, at this point, where V is V max, and the mesh size is the mesh size this point too, okay? And what you see here is that if indeed this scaling behavior is correct, I would expect that this time constant, okay, this decay time constant will scale with the viscosity of the medium. And we did it. So we changed the viscosity of the medium. You can add, for example, glycerol or whatever you want, and you really see that things scale. So this scaling behavior seems to characterize the behavior of the system, at least in the decay part. So we'll come back to the growing part in a few seconds. Okay, so this is what I said. Okay, so this was a top view, but now let's take a look from the side view. So in fact, here you see the three directions, I would say. So this is the top view. Okay, this is a cut in here. Okay, so from the side view, and here from the other side view. And what we found, okay, is that these gel sheets, they buckle. So they contract, and at a certain point they start to buckle. And the buckling really, so this is the phase of contraction, and really the buckling occurs at the periphery, okay, of the gel. So let's take a deeper look at that. So let's take a look at how it happens. So what I brought here is in fact a side view of the gel during contraction. And what we see that initially the gel really gets thinner and thinner. Merde, sorry. It should have, I mean, should be polite. The gel gets thinner and thinner, okay, and only then, after it gets at a certain thickness, it starts to contract laterally. So we have a thinning process and then lateral contraction. And what we see here, so what we did here is we plot the thickness as a function of time. So this is the small e, okay, the thickness as a function of time. And here, at this point, the lateral contraction of the gel that I've shown you before starts. So what you see here is that during this contraction process, the thickness doesn't change too much. I mean, by 10% at most. And after reaching a certain size, the system will start to buckle. Okay, here as I've shown you. Okay, so this is the process. And one of the things that you really have to note is that during this thinning process, lateral contraction is almost not important. It really starts afterwards. But the fact that the thickness doesn't change much after the system really shrinks by 90% tells you that the Poisson ratio here is 0. So assuming that volume is kept is not relevant. Okay, it's not true. So the system really is maintaining its thickness despite the fact that it's really contracting enormously. Okay. So as I said before, one of the things that we realized is that in order to generate this buckling instability, the density gradient, okay, so we have density gradients that form in the periphery persist. This is the first thing. The second thing that we also realize is that if you take a look at this plot, also what you see here are in fact three places, here, here, and here. So this is the center of the gel. This is the periphery. This is a point that we chose at a certain place in between them that we keep constant. One of the things that we realize is, of course, that the periphery really starts to contract before the other places. So as I told you, contraction started at the periphery. But, and the rest follows. But one of the things that you also realize is that the density, again, this is the density of the gel, really reaches a maximal value. And afterwards, although it reaches a certain maximal value, the rest of the gel continues to contract. So basically what you see here is a system that starts contracting, densifies, okay? The system continues contracting the center because it can continue. It is not dense enough. And eventually, the fact that you have this mismatch between the contraction process in the center and the fact that the periphery is very dense and cannot follow up is probably one of the reasons why we have this type of buckling. Okay, so this is an effective differential shrinking process, okay, but the initial formation of these gradients is spontaneous and is related to the fact that the system contracts from the boundary. And the reason it contracts from the boundary is because motors are, the boundary is the only place where the forces generated by the motors are not balanced because you have nothing in the outer part. that really balances them. So this is why it starts there. Afterwards, it's propagate. This is the way we see it, okay? So what do we get? So in the end, we get these type of structures. So these are very nice structures. So you can see it from the top here. And these are these buckling wavelengths. You can characterize the buckling wavelength of the structure, okay, and the thickness. And what we found is that we can generate sheets with different thicknesses simply by choosing chambers, okay, with the same lateral dimensions but increasing thicknesses. Okay, so changing the chamber height, initial chamber height, generate gels with thicker thicknesses, final thicknesses, and this eventually give rise to sheets with thicker thicknesses, and the wavelengths, what we found is scale with the thickness linearly. Okay? So this is the first thing that we get. Okay. So now about fluid flow. So obviously the system is in water, right? Or in solution. And you contract so efficiently, water has to be squeezed. Okay, so you have to really expel the water outside. So initially, one of the things that we imagine is that okay, so water molecules are not moving. Okay, so everything contracts and the water is standing. This gives rise, of course, also to friction. Okay, this is not a problem. We still have this friction effect. But now, what we realize is that, in fact, if you do some conservation of the overall density of the system, gel plus water, obviously water has to be expelled. So I know that the movie is not superb, but still, what we did here, yes. Okay, so let's try it again. Okay. It does whatever it wants. Okay, so we'll explain what we see. So what we did is okay. So what we see here is the following. So we have a gel. So this is the actin. The resolution looks really lousy, right? You have to imagine that we have the size of these gel are typically 1.5 millimeters. Okay, so big ones. So basically, we look with a loop. Yes, as if it's very, very low objectives lengths. And what we did is we added fluorescent molecules. Okay, but they have to be small enough to be able to penetrate the gel, of course, and to be visualized. So I know it's not very easy, and the movie is not working. So we'll leave it here. But I will just show you a few examples. So you can follow beads and measure their trajectory, okay, and the velocity of these beads. And what you see here is typical velocity of beads. So let's say this one that is going out. Globally, they are moving in the radial dimension, globally. And what you see that if this is, okay, the velocity of the boundary of the gel, this is the blue one. And this is typically 7 to 8 micron per second. You see the velocity of the fluid that is getting outside can reach something like 20 times higher. So it is a huge expansion of water outside. I will get back to it a little bit later. So what you see that we have an increase in the velocity. This is a point where we reach, this small bead reaches the gel boundary. And what you see that the velocity is still not zero outside. So the fluid continue to move outside, although the small bead got out. But one of the things that you realize is that the velocity decreases before it approaches the boundary. So we have a gel that contracts. Okay, obviously the boundary is denser because I told you that everything densifies the boundary. So it could be two things. Either the density of the gel is too high, such the mesh size is a little bit small, such that the beads are, getting stuck or getting a little bit hindered by the mesh, not not stopping of course, completely. But obviously, we have something that prevent them to continue to increase their velocity. Also, we would expect it to happen. And it happens for all the bits that we checked. Okay, so very high velocity of the expelled the solvent. What I should say is that at the end of the process, when everything slows down, because as I told you, you have an increase in velocity and decrease, at the end of the decay part, where everything slows down, so you can really measure the fluctuations of the bead, what you see that the velocity of the bead really fluctuates inside this. And one of the things that we notice that the low velocities, okay, the very low velocities are places where we are very close to the meshes, and the highest velocity of the solvent is in the center of this pore. So really, we have a measure to show that the system is porous, and the solvent really follows the structure of the gel. But you can really see it only when everything is a little bit slowed down. Okay, so one of the things that we wanted to do is at least to understand a little bit what is going on. And I would say that we can explain maybe 10% of what we saw in the experiment. So what we did is the following. So the first thing that we wanted to understand is really the role of the water flow, okay, and the gel contractility on the contraction process. So obviously, in order to do so, we had to take into account both the flow, okay, so we use mass conservation. We take into account both the flow, the displacement field of the gel, and the solvent velocity, and couple the two of them. And obviously, the velocity are related one to the other, because what we maintain really is that the total density of matter in a given volume area is kept constant. So obviously, the solvent flow is generated by the fact that the system contracts. Yeah. We took also into account the fact that we have force balance. We have at a low Reynolds number system, so obviously the total force is equal to 0. So we have balance of forces, which include the stresses, the elastic stresses in the gel, and the active force generated by the motor, and this is equal to the friction, which is the relative velocity between the gel contraction and the velocity of the solvent. And know that the active stress, I don't know if you are familiar with that, but it is proportional to the, I would say, the chemical potential, which is how much energy you get from the ATP, and it's a component ADP and PI. But this is a phenomenological constant, and this is Q. Q tells you what is the fraction of bound motors. And what you need to what you need to take into account is the fact that the motors, okay, get more and more attached the more stress they feel. And this is really important. This part is really important. Otherwise, you cannot really recover the increasing the linear phase. Okay. So what we found is that basically we can generate a flow. So this is the flow of water outside the gel as a function of time. What you see here is the contraction velocity of the gel. So we start in the boundary, it increases and then decreases. Clearly you see that the gradient is not linear as we see in the experiments, okay, the gradient in the velocity. But we have places where it is, but not really at the boundary. And what we do recover basically is the edge contraction velocity. We have an increase and a decrease. Really, it is really quantitative. So I mean, we don't have any fitting parameter, which is really nice. One of the things that you notice here is that during the increase of the velocity, we have really an increase. Again, this is the fraction of bound motor. You need an increase in the fraction of bound motor in order to generate increase in velocity, because you need more and more force. But then it saturates. And this decrease in velocity is really associated with effectively as if we had a contractile force that is applied from the outside and really contract a system which is elastic, and we have a fluid of solvents that's getting out. So this is what we can recapitulate. So I will just mention this, the work of Effie and Ran, because when we got this type of instability, I went to visit Iran and told you, I saw your paper, it looks similar, etc., etc. And one of the things that realized is that so one of the things that really is very different in our system than this artificial system. So this is a cross-link network of a polymer, and what you have here is a gradient of cross-links that you can generate in the system. So you impose gradients of density of contraction in the system, and the fact that you impose it can generate the different type of structures. So this is one type of structure which is really similar to what we get. So you get more contraction at the boundaries than in the center, and you can also generate domes-like structure if you generate inverse density gradient. But you need this in order to generate these systems. In our case, the system self-organize spontaneously simply by generating this density gradients in the beginning of the process. I will just finish by saying, yes, we can do domes, and we don't understand fully how they form. I mean, in terms of the dynamics, it's not trivial, but they do form, and we can find the conditions to do it. And of course, if the system is very, very thin, you generate, of course, different type of structure, which are essentially folding, but not contracting. And this is really something that we are looking for. So I will stop here and will say that this work was done by two students in collaboration with Carsten Cruz and Sam Safran. And we are trying now to understand really what is in a more, I would say, local manner, the formation inside the gel and the strain that are formed during the contraction process, because obviously what I've shown you is basically something at the frame of the laboratory and not of the gel itself. So thank you very much.